Bismillahirrahmanirrahim. Allahumma inni as'aluka rizqan tayyiban wa ilman nafi'an wa amalan mutaqabbalan. Today our talk is about mutations and DNA repair. With our previous knowledge and with our previous outcome of the lectures, which was focused on the nucleic acids, transcription, translation, I hope now it will be very easy for you to understand the mutations and DNA repair. So if you have any questions in the meanwhile, you can just post in the chat box, or you can even ask me later. But I would like to also say sorry to, I would like to apologize to few of you who actually contacted me in the last weekend basically to have some queries. I was too busy with some shifting of my house and all those things, so I was not having access to few of my stuff. So I was unable to reply. So inshallah, I will all the replies to you today, whoever sent me the message. So today we are going to focus our topic today. These are our learning outcomes for today's topic, and we will be starting with the basic issue of what basically are the reasons of the DNA mutations, and then we'll follow into the next levels of DNA repair. And we will be discussing is DNA mutation versus DNA damage. Are they the same thing or different thing? And secondly, if DNA damage is there, then the DNA repair is needed, or if the mutations are there, then only the DNA repair is needed. So we will be focusing all together into that direction. So now we have the impact of mutation in DNA on protein. So we have a mutation in the DNA. Option number one, we are having a mutation in the DNA, or we are having the mutation in the RNA. So whatever the mutations point or position is, it will finally affect the function of protein or the structure of protein. So what we have here, we got the like we do have our last slide in which we have the DNA to messenger RNA to protein, and then we said that now the current concept is we do have the DNA which is converted to tRNA, messenger RNA, or to ribosomal RNA, and then we got the protein out of it. Okay, now imagine if, for example, the whatever we have in the DNA, it is basically translated into the protein. That is what the whole concept of central dogma is. But what we are trying to say here now, a kind of new thing, which will be like if you change the DNA sequence, put it in a different way, if we change the DNA sequence, so in that case, the protein will also change. Why? Because the DNA sequence will change the codon, and this codon will change the amino acid which is linked with it during translation. So changing the DNA is basically changing the codon, and it will change the amino acid which will change the protein itself. So this is an example of a normal DNA in which we have GAG, and this is the coding strand, and we do have the template strand. We have the CTC, but in normal cases, this DNA will give rise to RNA, and this RNA will give rise to protein, which is the GLU amino acid. So this is the normal protein. But example, in this specific gene, if instead of GAG you got a T here in the middle, the whole codon change. When the codon change, so instead of having GAG in the RNA, you will get GUG. So the entire focus will be changing, and this will give you valine, which is a mutated amino acid for this protein, and you will get the mutant protein. This is one of the very common example which is normally given, for example, in case of sickle cell. So sickle cell, we have the mutation in which the GLU will be changed to VAL, that is amino acid, because of the mutation in the DNA. So this is how the different mutations in the DNA basically they affect your structure of the protein, your function of the protein. So now we are saying that if you change the codon, you actually are changing the amino acid. So affect mechanism, transcription, translation, expression of gene will be there. Imagine if the same mutation is in the promoter region. We go back to the basic concept. We have the gene and we have the promoter regions before. I mentioned previously we have one promoter, one gene in eukaryotes, means there is only one gene which is transcribed by one promoter. But in case of bacteria, for example, there will be 3 to 4 genes which actually exist in the form of group, and they are transcribed by a single promoter. So this group of genes in bacteria is called operon. I'm trying to answer a few of the questions I received through the WhatsApp so that you can align back with the topic. So in both the cases, if there is a mutation in the promoter region, not in the gene, if there is a mutation in the promoter region of the gene, what will happen? Remember, we have mentioned that we have the transcriptional machinery in which different transcriptional factor will come and bind to the promoter region. So these factors will not come and bind because of those mutation, and even then the transcription of that specific gene will be affected. So this is like how the impact of the DNA is. It could be on the basically the on the gene itself, in which the transcriptional is working very well, translation is working very well, but you incorporate a mutated amino acid or mutated DNA inside your new product. But if you have a no mutation within the gene, but it is upstream in the promoter region, in that case, what will happen? The transcription and translation and even expression of the gene will be affected. Now we do have a basic concept. This is very important for all of you to understand. We do have the genes. You please remember this triangle in your life. We have the genes and how these genes affect your phenotype. How these genes they affect your phenotype is all studied in classical genetics. Okay, now what we have studied just now in the previous slide, we are trying to say, for example, if physiologically something, if not physiologically, pathologically something happened and the mutations are induced in the DNA of a person, either congenitally, for example, from the parents to the offspring, or it happened because of some natural reasons, chemical reasons, which actually induce the new mutations in the DNA, in the maybe somatic cells or the gametes. So in both of these scenarios, this is a generalized scenario. What if we put in a different way, like we want to induce a mutation ourselves in some specific gene, in some specific organism, or in some specific product? Example, we want to make a new insulin. This insulin will be having no insulin resistance. I'm just giving you one example. It will be there, but I'm giving you example to teach you. So we want a gene which, if you use, for example, on your to make a new product, and this product can make your skin very nice. So all these things can be happen within the laboratory. We normally don't need the human body for that. We can use a cell culture. We can plant it in different kind of bacteria. So when we are inducing these all kind of mutations, it has to be understood like we are using it through different platforms. So what we are trying to refer here is that genes, the behavior of genes and how they act on the phenotype is all studied in classical genetics. But if you want to study the effect of proteins on the phenotype, it is all studied in biochemistry. But how the genes affect the proteins, the base, it is under a topic of molecular biology. So what happened? Something very important. The if this molecular biology, this base basically, which is the base of biochemistry and genetics, it can tell you how the phenotypes can be achieved by changing the genetic code. Like how can you achieve a specific physical property by changing the genetic code? And then this whole thing is actually done under the scope of genetic engineering. So molecular biology, if you want to achieve something new, for example, a genetic modified organism, genetic modified gene, genetic modified protein, everything is done under genetic engineering. And then if you want this specific example, you have developed a new vaccine. So this vaccine was developed you by a specific for a specific bacteria, but you have used live attenuated vaccine, or you have used a mutated bacteria. Make it very easy. You have used a mutated bacteria, And this bacteria you found when you use in animal models, it is very helpful to evoke the immunity against that specific bacteria in real time scenario. So what you're going to do, this is all done under genetic engineering and molecular biology. But now you want to make a lot of copies of this bacteria to be available for the mankind. So this whole thing is done under biotechnology, making something at a lab scale and then bringing it to the bigger scale. It's called biotechnology, so that people are using it all over the world. So you have to make sure very nicely what the first part in which we are talking about the disease, all those kind of things, mutations, all those kind of things, it is related to human diseases on one aspect. But you as a doctor in the future, you also need to target the molecular biology, genetic engineering, and biotechnology. Because if you will be not there, only the scientists will be there. They are not going to end up in a proper products and inventions for the purpose of serving humankind. Okay, well, now what are mutations? So we now, based on our last slide, now we know that the mutations are not just bad. Sometimes the mutations could be good also in terms of developing, the genetic modified organisms and for the purpose of biotechnology. So we go to what are the mutations? Mutation is a change in the genetic material at the molecular level. So remember, we have different levels. We talk about the cellular level in which we talk about the cells. Then we talk about the cytogenetics level. What actually it means the genetics of the cells. Means we are talking at the chromosomal level, that we have the 46 chromosome. We are talking about the chromosome level as the cytogenetics. When we talk about the genetic, or don't want to talk about the gene level, it will be talking about the genes. Within the genes, we have the exons and introns. We're talking at that level, like how many, maybe one exon or intron is deleted. This is also a mutation. The third level is we're going to talk about the molecular level, ATGC. Within the genes, ATGC, any of them is mutated. So mutation is the change in the genetic material at the molecular level. Mutant is an organism that exhibits a novel phenotype, or this mutant could be a gene, it could be a protein also, could be an organism also. So it's an organism that exhibits a novel phenotype. They act as a source of genetic variability required for evolution. Some people assume that in the evolution we need a lot of mutations, but remember, a mutation, the rate of mutation is not that simple. It appears over generation and generation. So yes, the environment has a lot of effect. Remember last time we gave the example of epigenetics. Do you remember what is epigenetics? We give an example saying there was a boy who went to the doctor and he said I got a high blood pressure. If you remember that joke. So in that joke we said that anything which is not coming from mama papa but from other sources which can affect the expression of a gene or expression of a level of the protein, that is called epigenetic. So the genetic variability during evolution is not just the phenomena of the of the genetic transfer, but also the phenomena of the environmental impact. Okay, now we go to the classification mutation in vivo within the body. The first one is the spontaneous versus induced mutation. Number two is the gametic versus somatic mutations. Number three is lethal versus conditional mutation. So these are six different terms which you need to understand very clearly for the future. I have excluded some very difficult terms which could be for the in vitro studies, and which are very more specific towards the scientists rather than the clinical impact. So we just pick what is needed for us for timing. So we do have the spontaneous versus induced, then we have the gametic versus somatic, then number three, lethal or conditional mutations. So spontaneous mutations are those that happen naturally. There is no specific agents are associated with their occurrence, and they're generally assumed to be random changes in the nucleotide sequence of the genes. Then we do have the induced mutation. These are the mutations, those that results from the influence of any artificial factor. There are various forms of radiations, a wide spectrum of chemical agents, And then the biological agents. We have a very good example of the corona now. We said every time corona goes to any human, and then it comes out mutated. Yes, but remember, it is also playing with your own RNA and your own DNA. So it can also, bring in some mutations which were previously not in your cells. So then we have the gametic versus somatic mutations. Mutation arising in somatic cells are not transmitted to the future generations. So, but the mutation in the gametes or gamete forming tissues are of greater significance because they are transmitted to offspring as a part of the germline. So we normally consider them to be either dominant, either recessive or X-linked. Currently, they're not a part of our topic because it's more on genetics. We are not talking about genetics is a totally different, like I mentioned in the triangle. So genetic is a different topic. Currently, we are focusing through the molecular biology impact. So the dominancy, you know what is dominant? Anything which appear in a particular generation, but it's not in the other generations. The recessive is something which is not appeared in the form of the dominant alleles, and then X-link is which is related to your specific what we call the X chromosome. So then we have the lethal versus conditional mutations. Mutations may interrupt a process that is essential to the survival of the organism. In this case, it is referred to as the lethal mutation. For example, in case of there are some specific genes which are basically involved in during especially during the gamete formation. If any of these mutate genes are mutated, or example, if these genes are not transcribed because of some mutations, then normally the what we call your embryo will not be developed and it will just lose the life. And then conditional mutation is present in the genome of an organism, but it is expressed and can be detected only under certain conditions. Sometimes what happens, there is a gene and this gene is working very well in a person, but with the late onset, when the person reach 40 plus 45 plus, those genes stop working that efficiently. Remember, we do have the concept of topoisomerases, which we did, telomeres and telomerases we discussed is kind of assumed to be a kind of conditional mutation, also sometimes in few of the books. So these conditional mutations were present, but when you change condition, environmental condition, food conditions, age, many things, so these mutations start appearing and showing their effects. But in case of bacteria, there are few example of bacteria which is growing very well in the absence of sucrose. But there is a gene called SEC-B, for example. If the bacteria is having that gene, if you put this bacteria and grow this bacteria in the presence of sucrose, this bacteria will die because the SEC-B gene will destroy the whole replication mechanisms of that bacteria. Well, now what we have next is the types of mutation in vitro. We discussed about in vivo and now we go to in vitro. As per need, we can induce mutation. We have just discussed that if we need we want to induce a mutation to develop a new organism or to develop something more stronger which can be used for the mankind, we can induce the mutations. So most common are frame shift, based and point mutations. So then missense and nonsense mutation. We're going to discuss all of them. So we have the frame shift, point mutation, missense, and nonsense mutations. So now something need to remember, there's a level of mutation. So maybe it's a change in the chromosome number and structure. Point mutation is that a specific nucleotide in a gene is there. Insertion mutations insert fragment of the DNA. Deletion mutation is you deleted a fragment of DNA or specific genes. Okay, so till now, do you have any questions? If you have any questions, please you can ask me. If no, I can just move on.
Prof,
Yes.
sorry, I'm not very clear about the conditional mutation.
Conditional mutation. Okay. Make it very simple. The gene is already there. Okay. For example, the gene is already there, but what happened? Put in a different way. What happened? This gene, for example, this gene, you want this gene to be expressed under specific condition. Like any reaction to occur, any protein to be formed, there is a specific reasons for that for the gene to make it more easy for you. Example, heat shock proteins we discussed in last lecture. So normally these proteins are not activated. The genes involved in heat shock proteins are not activated unless and until you have a heat shock. Heat shock in bacteria is different, in humans is different. I'll give you a simple example. For example, you have been to some place which is very cold, and you once you come back to your normal temperature, like you come back to again a hot place. So what will happen? There are few of the genes which are activated at a very cold temperature. They will either turn down, means they will not stop expressing again, or either they will become more advanced because they still want to express. So there's some specific diseases. Example, we have sickle sick what we call sick serum diseases. There's some other diseases in which specific antibodies are formed against these specific proteins also. So make it in an easy way. There are few genes which are actually in a specific condition they express themselves, and in other conditions they don't express themselves. So these are called conditional mutations, or that specific mutation that only will work when only that condition is there. For example, some of the mutations which are in epilepsy. Okay, so these mutations sometimes they only start working, or they start getting energized at a level of the nervous system only in a specific condition. Are you okay now? I try to make it very simple because I'm so worried. In my actual lecture, there's so many of examples, but because with you all, try to make it very simple for the concept. Are you
Understood,
okay?
Prof.
Can I move on? Uh, Understood?
Yes.
Okay, Alhamdulillah. So now we have the level of mutations. We move. Okay, now we go move on to the next part, which is a DNA damage versus DNA mutation. Okay, now what normally happens? There is a if a mutation is induced, or if the mutation is there inside a person. Put in a different way, we are not talking about the mutations at the gametes level. We are talking example in the somatic cells or the gametic cells, but the person is already born as adult, for example. So these mutations, if the mutation is caused and this mutation will not be repaired, because in our body we also have the repair system for these mutations. If that system, the repair system will be failed and it will not repair that mutation, then what will happen? Then the DNA will be damaged. So don't confuse. Sometimes we assume that DNA damage is equal to the DNA mutation. They're totally two different things. So there is a mutation which can give rise to DNA damage. Why? Because of the failure of DNA repair system. But if your DNA repair system is working efficiently, then the DNA damage will not occur. Okay, so what we have now is, so if you have on the right side, if you see, sorry, in the on your screen, maybe on the left side, if the DNA damage and the DNA repair is equal, if they're equal, then the genetic stability will always be there. But if you have the DNA damage, if you have a DNA damage, but you have a defective, incomplete DNA repair, then it will be genetic instability, because your genetic material, which is your DNA, is now unstable, and you are going to get those mutations in your body, in your somatic cells, in your gametic cells. So what will happen now? For few of the example cancer, hereditary diseases, they will start appearing in your generations or even in yourself. Number one. Number two, there will be genetic divergence. We'll discuss about it in future lectures on genetics. So this is, I owe you for this genetic divergence. Currently, I'm not covering it because you will get confused. So this one will inshallah focus in some other topic for sure, inshallah. But we have discussed a little bit of I remember the wobbling effect, and we discussed few of these terms actually during our what we call in translation and transcription. But this is more generalized towards diseases, this term, rather than for the transcription and translation. So remember the concept is DNA repair is a key. towards DNA damage after getting a DNA mutation. Okay, now the molecular basis of mutation. Mutations alter the nucleotide sequence of a gene in several ways. The substitution of one base pair for another, for example, A for T, the deletion of one of few base pair, that is AT to example GC. Okay, then the one of the reason is the tautomeric shift. We have discussed this part in the last lecture. So what actually is happening here, if you see what the common form of these are the common form. Example, this is a common form of thiamine. This is a keto form which is common in us, but the rare form is this one, the enol form, for example. So this is a tautomeric shift. Similarly, we have for the cytosine, we have the amino form which is changed to amino form, and then we have the adenine amino form to amino form, and then we have the guanine which is keto form to enol form. So that is how basically the tautomeric shift can actually give rise to a new base pair in the form it's considered to be a kind of mismatching or mutations in the future. So this is one example in which, for example, normally the cytosine, for example, versus adenine bind to each other. Normally, remember the adenine normally binds with thiamine. A normally bind with T. But normally, this is another example in which cytosine rare amino form. We go back to our previous slide. Cytosine is basically, if you see here, cytosine is basically normally amino form. So what happens if this one is changed to amino form? So then what we get an example, instead of binding with G, this cytosine is going to bind with A. So here instead of binding with G, it will bind with A. So this is a mismatching. Okay, similarly, normally thiamine binds to A, but what happens when the guanine rare and enol form normally bind with thiamine? So instead of T binding with A, now T is binding with G. So this is how the small tautomeric shift can actually affect a mismatching, and it can change the entire sequence of the DNA, unless and until if this is not repaired by some specific proofreading enzymes in our body. We're going to discuss about it. Okay, now we have the mutation. We similar example. We have the mutation caused by tautomeric shift. So if you see here, this is the parental DNA, and when it got replicated, here we got the mutation. Instead of AT, we got GT. So now this is their replicating. This is the first generation progeny, and now this will finally appear in one of the generation at the end. So this is how slowly this mutation will start appearing in the generation. And now, if any of the individual is having this mutation, it's going to have some disease. Number two option is the first one we said, okay, please remember, these are not the types of mutation. We are talking about the what are the bases, what are the reasons of mutation. The first reason of mutation we say is a tautomeric shift. Number two is the base substitution. What happened in base substitution? There are two types, either it's a transition or it's a transversion. If it's a transition, it replaces pyrimidine with another pyrimidine or purine for another purine. So easy if it is if it is pyrimidine to pyrimidine or purine to purine, it's called transition. But if it replaces pyrimidine with purine or purine with pyrimidine, this is transversion. So these we have just explained it here. So this key is pyrimidine to pyrimidine, purine to purine is transition. Transversion is pyrimidine to purine or purine to pyrimidine. Okay, so what we have next is the frame shift mutations, alteration of the open reading frame. Okay, something I want you to understand very well now. Otherwise, you will be always confused. I think maybe some of you have already gone through this part before in your previous classes. So if you see here at the top, I'm writing something. You have ATG, EGC, CGT, and CGT, CAC. Okay, can you all see this, the text I've just added here? Okay, so now what we want here now, we want to do something new. I want to now delete a part here. I want a new sequence in which this A is deleted. So we assume after A is deleted, the first A in the start, the first A, I'm so sorry, you have to write again. So we have ATG, TGC, CGT, CGT, TGA. So now what we have here, now I want to delete the first A. Are you all okay? Are you understanding? So we have this, we 1, 2, 3, 4, 5 codons we have. I want to delete the first codon. In the first codon, I'm deleting only A. So what we get here, we will get T. So now what we got here, T, G. Remember we have deleted A. Now the codon is shifted. So next T will become with this T. T. And then the codon will be shifting. We got G, C, C. And then we got G, T, G, C, T, T, G, A. So this is our new sequence because the A in the previous sequence, oh, mashallah, A in the previous sequence was deleted. Did you got it or not? Because I think I don't know what's something happening with this WebEx. It again got. Let me try again to. I don't know. Sorry. Okay. So now we go to the next example. Now we go to the next example. We got ATG, ATG, CGT, GGT, and then GCT. Now, in this example, what I want to delete the first two. So the sequence will be G, CG, T, GG, T, G, C, and T. Okay, from there we delete another G from the original sequence, so it will become the whole codon is gone. So C, G, T, and onward it will be same. So now in these all examples, what I want you to understand here is if you see in this example overall, so if you see the first one, if A is gone or two base pairs are deleted from the left side. So what happened with this deletion? The frame, that is frame, what actually the frame is your codon. The whole frame is shifted to the right side basically. So what happened? G will get another few bases from the other codon, so it always will be appear in the form of the codon. So every time the codon change, your amino acid sequence which it will be coding will be changed. So if you delete even a single base pair, what will happen? The whole codon will be shifted to the right actually. Sorry, to the left. It will be shifted to the left. So always remember the trick is if example we put now last example here to make you understand. I put the same sequence now in a different way. I want to add one more sequence, so I add T to the previous sequence. A, T, G, C, G, T, G, G, E, C, and T. If you see here, I have added only a T here. So this adding the T here, which has shifted all the codon to the right side. So the amino acid previously, which was ATG, now it become TAT. So the amino acid will be, the nucleotide codon is changed and we'll get a different amino acid. So in this example, we have actually inserted or we have added a new base pair. It could be one base, it could be two base, it could be three, it could be the whole exon, it could be as much as you want. So in this case, it can happen in vitro. Sometimes it do happen in vivo also. But normally we are now discussing in vitro if we are inducing the mutation. So we have the insertion, we have the deletion. But what happened? Both of these insertion and deletion, they basically shift your frame, shift your codon. Okay, so that's why it says the alteration of the open reading frame. What is open reading frame? Open reading frame is the whole the codon starting from start codon to the stop codon. This is called open reading frame. Okay, start codon to stop codon is all open reading frame, and then you can shift the frame, you can shift the code by adding or inserting the base pair. If you add anything, if you add a base pair, this is called plus mutations. If you delete it, it's called minus mutation. So if we add one base pair, it will call plus one frame shift mutation. If you delete one base pair, it will call minus one frame shift mutation. Are you all okay? Are you understanding me? Okay, Alhamdulillah. So this is how we actually induce the mutations, and these mutations will be appearing all together in your proteins basically. So what we have the DNA, which is the wild type, and this is the DNA in which we have the mutation here. So what happened, if you see here, this mutation basically actually shifted the whole codon, or it will give you a different protein. This is example, we are getting a different protein out of it. So because the whole thing is shifted. Okay, so this is how the mutation show their effect in the from the DNA into the protein basically. So looking at the amino acid sequence down there, the polypeptides are methionine, lysine, glycine, proline, threonine, etc. This one is totally different. Both of them become totally different from each other now. Okay, so we move on to the next part now. So mutation frequency, if somebody asks you, like, do you think the mutation occur in every generation? Do you think how the mutation, so there is a frequency of the mutation, like what is the level of, how frequent this mutation will occur? So frame shift, transition, transmission mutations are infrequent. Bacteria and phage, that the bacteriophage are, they have 10 raised to power minus 18 to 10 raised to power minus 10 per nucleotide pair per generation. So in one generation, this is a very, very low chance that the bacteria is going to have a mutation. In case of eukaryote, it's 10 raised to power minus 7 to 10 raised to power minus 9 per nucleotide pair per generation. It's the chance is so rare that the mutation will be there. Okay, but remember we have the silent mutations. For example, down there I've given you example. Remember we have the wobbling effect in tRNA. For example, UCU also give you serine, UCA, UCC, UCG, they also give you serine. In this specific case, if the mutation is silent, or put in a different way, if for example, the DNA is different, but it is giving, even after getting a mutation, it still give you the same amino acid. This kind of mutation is called silent mutation. Silent mutation, or in some specific cases, if this mutation can also give a problem to the diseases, okay, then this mutation is called variation also. The term variation is there. If example the silent mutation is giving any kind of disease condition or associated with a specific disease or character, then it's called variation. But that is too high expectation from you. Don't go to the variation for now. You just need to understand the silent mutation concept. So then we have the ionizing radiation causes changes in chromosome structure. So there are different ionizing radiations which can break chromosome and can cause deletion, duplication, inversion, and translocation. Okay, now this actually depends on the ionizing radiation. Basically, can destroy the bonds within your nucleotides. So these are different energy levels of those bonds, and if you see here, the CH bond is the strongest energy level as compared to CC and CN bonds. So overall, if you see the nucleotide, the bonds are very high in energy, and it's not that easy to break them. Okay, so what we have here is we have the mutagenesis by ultraviolet irradiation. Mutagenesis is basically generation of the mutation. So what happened when we have the UV light? Example, some people they are sleeping a lot under the sunlight. So what happened? There is a hydrolysis of cytosine to hydrate may cause mispairing during replication. So we have example, we have the cytosine basically, we have the cytosine nucleotide with the UV, and in the presence of water, it changed to cytosine hydrate. And then the other example is the cross-linking of adjacent thymine. You have T, there we have normally example in a 5 to 3 direction. We have example 2T are there. So what happened normally, they have a bonds with the nucleotides on the opposite strand. So what happened? This UV light, it breaks that bonds in the opposite strand of the nitrogenous base, and it make the bond between the two of the thymine basically nucleotides. So actually what it means, it make a small niche or small defect within the double-stranded DNA, and it create double bonds within the single same strand of the DNA. So this cross-linking of additional thymine form thymidine dimer. Dimer means two molecules in easy way, which blocks DNA replication and activates DNA repair and mechanisms. So this is examples of mutagenesis by the ultraviolet irradiations. So types of chemical mutagens, we can also use different chemicals. So chemicals that are mutagenic to both replicating and non-replicating DNA. Example, alkylating agents, and we have the nitrous acid. Chemicals that are only mutagenic to replicating DNA. Example, base analogs and acridine dyes. You please need to remember these terms: alkylating, nitrous, analogs, and acridine dyes. Just remember, we have no other choice. You have to, for the timing, remember by heart. Okay, so we have the chemical mutagens. We just explained now. We have example as the mustard gas. Then we have the ethyl methyl sulfonate. Then the ethyl ethane sulfonate. They all mutate the DNA. Then we have the base analogs. Okay, what are the base analogs? We are going to discuss in detail after this actually. But just remember, they are basically they come and they settle somewhere in the middle of the DNA, and then this when they settle in the middle of the DNA, this will give some kind of problems to the DNA itself. Are you all okay? Are you all following me? Alhamdulillah. Okay. Then we have the acridines. We have the 2,3-diamino acridine. The other name is proflavin. And then we have the amining agent, which is the nitrous acid, and we have the hydroxylating agent, which is the hydroxylamine. So from there, we can just proceed now. We have the alkylating agents. Chemicals that donate alkyl group to other molecules are called the alkylating agents. They induce transitions. Transition, now you remember what is transitions? Transversions, frame shifts, and chromosomal aberrations. And number two is alkylating agents or bases can change base pairing properties. For example, GC to AT can also activate error during a repair process. So when the repair process is going on, they can also induce error by interacting with the different steps of the repair process. So these are some examples of the alkylating agent. I think you need to remember one or two, which is easy for you. Okay, then we have the base analog that, for example, is a 5-bromouracil. There are similar structures incorporated into the DNA, increased frequency of mispairing. So we have one example here. This is bromouracil. Normally adenine bind to T. What happened in this case? This is bromouracil which comes and it bind with adenine. So what happened when it bind to adenine? The adenine which has to pair with T, now it is going to pair with bromouracil, and the bonding with the thymine is gone, and this will give you a problem in the DNA. Similarly, the bromouracil, we have the enol form, and normally guanine actually match with what we complement with cytosine, but in this case it will go for bromouracil enol form. So remember, this is one of a very important one, which is sometimes even asked during the exams. So remember this bromouracil in a very nice way. Don't forget it. Okay, then we have the nitrous acid causes oxidative deamination. Remember nitrous versus amino. So D, it causes deamination. So example in case of adenine, for example, in case of adenine, it changes adenine to hypoxanthine, and this hypoxanthine will mismatch with cytosine. Remember, the cytosine has to match basically with guanine, but now it's going to match with hypoxanthine, which is coming from adenine. Similarly, cytosine is basically because of the nitrous acid is converted to uracil, and this uracil instead of this uracil is going to have an issue with when it's going to bind with the adenine. Similarly, guanine will change to xanthine, and xanthine will be binding with cytosine to induce some mutations in the replication. So now we have the intercalation of an acridine dye causing frameshift mutation. So this is proflavin molecule is example one of the example of the acridine dye. So what happened? It just goes and incorporates in the DNA. Remember, the DNA, as we said, it's a helical strand. So it goes, it places itself like, like a parking your hand over another hand. Similarly, it go and it stack over the different bonds, and then when it stack over the different bonds, the DNA becomes so rigid, and Z, A, and all these the forms we have discussed in one of our previous classes, those forms could not be attained properly. So it's kind of the conformation chain, because this will not allow the DNA to bend properly. And if DNA is not bending properly, it means when the replication start, it will be not becoming the denatured or melted properly, and the enzymes and all the machinery of the transcription will not come and bind to those DNA molecules properly. So these proflavin molecules is an example of the dyes which are intercalating. Okay, then we have the hydroxylamine. Hydroxylamine is a hydroxylating agent. Hydroxylamine hydroxylates the amino group of cytosine and leads to GC to AT transitions. And then we have the term transposons. Please, I need to focus, I need all of you to focus. There's a term called transposons. Transposons are considered like jumping regions of DNA or repeats. These are the regions which can go from which can jump from one chromosome to the other chromosome. But funny, but this is what is happening in the genome. So, for example, our transposons from one gene actually jump. This is the transposon which jump from one gene and it jump to another gene. Now it comes to another gene here. So what happened when it become a part of gene? So remember, when this part will be jumping, it will be bringing its own ATGC, it will be bringing its own exon introns. If it is a very big size, normally it's small size. So when it will be bringing its own base pairs, on gene, on nucleotide bases, so they will become a part of that gene. And after becoming a part, the whole frame will be shifted. Additional codons will be added in this gene. But in the previous gene, the codons will be deleted. So minus frame shift will be up in the previous gene, and plus frame should be in the new gene. So this will change the whole, the protein function and structure, and maybe you're going to get inactivated, truncated, or non-functional protein. Inactive truncated is more like a non-functional protein. So this is again happening in the genome. Okay, now we have the expansion of trinucleotide repeats. Simple tandem repeats are repeated sequence of 1 to 6 nucleotide. Sometimes what happens is CGC, CAC, CTG, if they have to be example only one repeat, but sometimes they become more than one, maybe six or seven together in a genome. So when it happens, the codon is the frame is shifted, and then the protein will be truncated. We have some examples with the trinucleotide repeats, which are very dangerous, very lethal diseases. Example, Fragile X syndrome, Huntington disease, spinocerebellar ataxia. So these are all the diseases which are based on trinucleotide repeats. Okay, so remember, these codons become additional, and this will give you the disease. So this is a summary. We talk about the mutation, transitions, translations, frame mutation, and chromatin remodeling, which is more towards epigenetics, and which we say profilin, and which we change the conformation. So it's more moving towards epigenetics, because the DNA is not changing in those cases. Okay, now basic features of the process: mutation occur in all organisms from viruses to humans. They can occur spontaneous to be induced or by mutagenic agents. Mutation is usually a random, non-adaptive process. So accuracy of DNA replication machinery count a lot in the rate of mutation. If that DNA replication machinery is working properly with the proofreading activity in a nice way, then the mutation normally don't occur. Efficiency of the mechanism of the repair of damaged DNA is very important. If the DNA repair mechanism is not working properly, then it will also give you the mutation, which will be moving towards a DNA damage. And then with degree of exposure to the mutagenic agent, example, if it is in vitro and you're using ionizing radiations, you're using mutagenic chemicals, or all of those kind of things we have just discussed, then more higher of those concentration, and somebody is again and again exposed to that environment, then again the mutagenic these mutations can occur. We go back to the conditional mutations. Somebody asked me the question. So if you are basically evoking a genome towards a specific mutagenic agent again and again and again, the chances of the conditional mutation will be high. So types of DNA damage. Okay, this is something very tricky topic, but the most important thing I want you to grab now in this lecture is we're going to repeat some of them in the future also. But what I want you to grab now, the basic concept: what is the types? What are the types of DNA damage, and what are the types of DNA repair? Just try to grab the basic concept in the next 5 to 7 minutes, and then we'll finish this lecture. Okay, so normally what we have the deamination, depurination, and then we have TT and TC dimers. we have alkylation. Okay, so four basic type of DNA damages are there. If somebody asks you, please list the DNA damages, explain one of them. So this is the slide you have to always remember: deamination, depurination, TT and TC dimer, and alkylation. Okay, now these are all the same examples I've shown here. I want you to go through these slides when you are when you're going to read it again. So we have with this as examples: cytosine is deaminated to uracil, adenine is deaminated to hyperxanthine. We have already covered all of them in the previous slides. Similarly, we have the depuration, in which we have basically the guanine, which is depurinated, and we have some of the exposure to the UVs there in the induction, and we have the alkylation. So you have to go through all these slides. Okay, now type of DNA damage. Please, I need you to focus. Still, continue with the oxidative damage. We have the replication error. We have the double-stranded breaks, and sometimes the DNA is only damaged inside the single-stranded. Sometimes it's damaged in the double-stranded. So now something very important to understand. We are going to the DNA repair now. So by mechanism is the enzymatic correction of errors in DNA structure and sequence that protects genetic information against environmental damage and replication error. Remember this definition by heart: is the enzymatic correction of error in DNA structure and sequence that protects genetic information against environmental damage and replication error. So it will be epigenetic, it could be genetic, but the entire idea is enzymatic correction of errors in DNA structure. Okay, now the type of DNA repair could be error free or error prone. Error free, we have two types. We have the direct repair and we have excision repair. Error prone is SOS repair. So we'll discuss about them. Now, okay, again, error free is direct and excision. Error prone is SOS. So we talk about the type of DNA damage. So these are different types of DNA damage. If you can see, we have the double-stranded DNA and we have the mismatch. We have the mismatch. We have the double-stranded break here. This is the double-stranded break here. Mismatch. They are not matching properly. We have the CUD deamination. We have the AP side. Okay, so A purine or A pyrimidine side. One of them, the base is missing here. AP side is A pyridine, A pyrimidine side. We have the covalent X linked, and we have the thymidine dimer, and we have the SS break. So these are different kind of DNA damage which occur basically in case of DNA. So we talk about the direct DNA repair first. We talk about the direct DNA repair in which there is no need or there is no new DNA is required. This is a trick. There is no new DNA is required. So what happened? Remember the cyclobutane pyrimidine dimer photolyze. Just remember this cyclobutane pyrimidine. So what happened? Your DNA is there. There is a problem with your DNA. Sorry. So there is a problem with your DNA here. The mutation, the red one, the mutation is there. So what happened? This CPD photolysed bound to the dimer. It will come and it will bind here. And this is pink one is your basically the CPD, and the dimer just a small piece of DNA, which is a small piece of nucleotide, which will be used for the basically correcting this DNA. And then we have the absorption of light at 300 nanometer, and then reversal and release of enzyme. So the enzyme is released, and then you get the DNA without any mutation. The tricky part I need to remember is there is no DNA is required. CPD is involved, and is basically this enzyme basically work on a single strand, and it change this single strand to the normal strand. Second option is excision repair. It says undamaged DNA strand provide DNA template. Remember we have one strand which is not damaged, so it will provide you the DNA. So mechanism of repair of environmental damage to one strand of DNA: loss of purines due to thermal fluctuation resulting in formation of pyramid dimers by UV radiation. For example, TT we just discussed, and the site of damage is recognized, excised by endonucleases. Okay, I want you to remember the name of these enzymes. Endonucleases. The corrected sequence is copied from the complementary strand by a polymerase. The end of this correct sequence are joined to the rest of the strand by the ligase. So first the endonucleases will come. Please try to understand. First the endonuclease will come. They will cut that strand. They will excise and cut the mutated strand. and when the mutated strand is cut off or chew off, number two, the polymerase, like remember we have the DNA polymerase, the polymerase will come and it will actually add the missing strand now. So first step, endonuclease will basically remove that mutated bases. Number two, the polymerase will come and it will produce that bases. Remember, it used the template from the other strand, just like in the replication we have discussed in the previous lecture. So it will use the template strand. It will use the coding strand or the template strand as a what we call to add the sequences at the site of missing sequences, like where endonucleases act. Number three, the ligase will come and it will join back all the strand by creating the phosphodiester linkage and all those kind of things. So remember, three enzymes: endonuclease, number two polymerase, number three ligase. Are you all okay? Can you see the difference between excision repair and the direct repair? So we have the direct repair, we have the excision repair. So now both of them, we said, we go back to one of our slide here. We just mentioned both of these mechanisms as error free. There will be no error because they are highly enzymatic. Okay, so they are error free, both of these mechanisms. And now we are going to the next one, is called SOS repair. SOS stands basically for so many things, but make it easy. This actually I found the easiest one was written for all of you on Wikipedia. To be very honest, if you look for the source, sometimes it's important. So I found the one they have put on Wikipedia. I just take a little bit of it from there because it's a very complex process. But what we want to focus in current lecture is that you at least get the basic concept of this pathways. So SOS response is a global response to DNA damage in which the cell cycle is arrested. Do you know what is the different phases of cell cycle? If no, these are different phases: G1, S, G2, M, and GO. So there are different phases of cell cycle. So what happened when the DNA is not forming? Example, there is a block in the DNA formation, or DNA is in the process of repairing after the mutagenesis. What happened? One of these phases of growth, they got arrested or they got blocked. So when they got blocked, so it will also give you the issue with the DNA damage. So remember, again, SOS response is a global response to the DNA damage in which the cell cycle is arrested and DNA repair and mutagenesis are induced. Okay, so what the most important gene involved is the REC-A gene. REC-A is recombination. So REC-A gene is involved, and make it more easy. So this REC-BCD is a repressor. What is a repressor? Repressor is a molecule, like few minutes back somebody asked me with again with the conditional term, So the REC-B gene is actually a repressor which will not allow the REC-A to act. This REC-B has to be removed from this stand. Once this REC-B is removed from this stand, only then the REC-A can come and it can bind at this position to start the next processes. So this REC-B has to be removed to continue the process. Okay, so this is how basically the REC-A is involved in the SOS repair, but in this is the error prone. The first two are error free. So this is how the generalized two different repair pathways. In the first we have two types, and the second we have only one type. So then I think that is all from today, except this last slide, I think. So we have the DNA repair defects, causes, and diseases. I want all of you at least to focus on the first two columns only. So the first two columns, we have the disease, example, prevention of point, mutation, insertion, and deletions. We have the hereditary non-polyposis colorectal cancer. It's a DNA mismatch repair. Xeroderma pigmentosum, it is nucleotide excision repair. And then for the repair or double-stranded DNA breaks, we go to this next disease. We have Bloom syndrome, which is the repair of double-stranded breaks by homologous recombination. Fanconi anemia, which is basically the repair of double-stranded breaks by homologous recombination. Hereditary breast cancer, BRCA1 and BRCA2 deficiency is a repair of double-stranded breaks by homologous recombination. So this is breast cancer. These are other anemias. These are all very vital diseases. You want to study all of them, you can. But because the issue is, I didn't go to detail of these diseases for now, because I am 100% sure you need to go through homologous recombination before that. There's so many terms we need to go through in genetics before we bring in these terms. So currently we just focus on just a basic concept, and then we move on from there. So I think that is our last slide, and that is all from me. Thank you so much for all of you for being attentive and so patient. I know it's not easy lectures for all of you, and you're trying to digest as much as possible. If you have any questions, please you can stay back, and if you have no questions, you can leave the meeting. I will not mind, inshallah, because I think you're going to have a next class in the next few minutes, so you can have some rest.
Thank you.